talked about different tips for effective communication. Especially, I discussed with you that how one could make effective oral presentations, as well as gave few tips for the writing. So in that way, I'm hoping that you are quite comfortable overall for the various presentation tips, right? Um Before I continue, can somebody confirm me that my slides, audio, visual, everything is okay?
Yes, sir, everything is okay.
Yes, sir.
All right, very good. So we're continuing from that, and today I am going to talk to you about rather a story of cloning, and I'll try to give you a perspective that how research can have different type of twists and turns. The basic science research, how challenging it could be, and different ways different scientists who have tried to investigate these problems have seen lot of the rise and fall in the area of research. So we'll talk about animal cloning and stem cell cloning. Also, I'll talk to you about some of the cell reprogramming studies and how some of the ethical issues were brought while some of these research were happening. So, of course, it's not a biology class, so I'm not going to talk in detail about these concepts. Of course, I'll be happy to take any question at the end if you want to learn more about it. But just to set up the stage for everybody to know what the topic is about, as per the Human Genetics Advisory Commission, the cloning is a process of producing a cell or the organism which is having same nuclear genome as the another cell or organism. So in some way, you are producing the photocopies of living cell, which is a genetic duplicate. And that was kind of a very elegant concept, which actually started as early as in 1930s. So 1938, the first time the animal cloning experiments were actually started when Hans Spemann, he showed that this kind of concept is possible by fusing an embryo with an egg cell. But actual success of cloning an animal happened in 1962 when scientist Dr. John B. Gurdon, he first time cloned frog. But of course, still no mammal and the large animals were cloned. After several years in 1986, Dr. Steen Willadsen, he cloned a live lamb from the mature sheep embryo cells. 1994 was the calves were cloned by Dr. Neal. And 1996, Dr. Ian Wilmot of Roslin Institute, he first time cloned a sheep, Dolly. And I'm sure you would have heard about Dolly cloning in different contexts. So this was the first large cloned animal from somatic cell. And this was quite elegant experiment to say because sheep is of such a huge commercial requirement. Plus, it is kind of very large mammal. So this was quite successful experiment, which really set up a big stage for a lot of cloning field in general. So what he did, he took one sheep and took the memory cells, did some, the cell cycle arrestation of these cells so that they can do dedifferentiation. And from another sheep, he took one egg cell from which, now he has removed the nucleus. So now it got an envelope which is having no nucleus or anucleated cells, egg cell. So took the memory cell from the sheep 1 and took the nucleated egg cell from the sheep 2, fused them with the electroporation and different tricks. After many attempts, one of those really went well inside the nucleus, which formed the embryo. And then now this was implanted in the third sheep, which became the surrogate mother. And that's where the embryonic development happened. And the lamb Dolly, which was the clone of this particular sheep, was actually the first large clone animal was produced from the somatic cells. It became quite landmark study, and I must say that I was very fascinated that time with this kind of work going on. I was graduating that time, but when I reached to Harvard after my postdoctorate work, it was just so fascinating to meet Dr. Ian Wilmot over there, and we somehow gave a talk in the same conference on different topics. Of course, I work in the proteomics, not in the same field, but it was really fascinating to get his perspective and see his vision for this field. So it was very, very much like you see science fantasy, and then all of a sudden things starts coming into the play, and then you meet the scientists who have done this kind of work. So I'll just show you this news clip. If you're not aware of the significance of the Dolly cloning, you will listen to this particular clip.
The headline on the other major news story today, to which we intend to devote some time, is very simple. Hello, Dolly. It's a very long time since a science story on Sunday made such waves on Monday, but with good reason. Dolly is the name of the first mammal, yes, a sheep, ever to be born as the result of cloning. Cloned from an adult sheep in a laboratory. Dolly is doing fine, thank you. She is 7 months old already. And from the scientific community to the water cooler, we human beings have had a future, or seen a future at least, that had only been imagined up until now. If a lamb, why not a man? Our first report from ABC's John McKenzie.
Dolly is without question a normal, healthy, genetic marvel. She's an identical copy of an older sheep.
People expected that maybe at one time, sometime it would happen, but maybe we've been a little bit quicker than they expected.
Dr. Ian Wilmot and his team of Scottish researchers began with a 6-year-old sheep. They took a single cell from her mammary gland, including its nucleus, the command center containing all the genes, the DNA, that determine the sheep's individual physical characteristics: eyes, nose, hair. Then they took an egg from a second sheep, removed its nucleus, and replaced it with a cell from that first sheep. The egg and its new nucleus then fused together, turning into an embryo, but with all the genetic characteristics of the first sheep and none of the second. In effect, a carbon copy, an exact duplicate of the first. All that was left was to put this developing embryo into a third sheep, a host sheep, which carried it to term.
The key to this experiment was to induce the genetic information of a very specialized cell from an adult, in this case a mammary gland cell, to go.
All right, so I hope you got a glimpse of how the Dolly was cloned. But let me go back a little bit for one of the landmark study by Dr. John B. Gurdon. In fact, he was the first scientist in 1962 who showed that animal cloning is possible. And then he used, for that experiment, he used for the frog egg cells. And then he showed that, it can be turned into any specialized cells. So what he did, he took, again, like, what Dolly cloning was done quite late in 1997, but quite early in the days in 1960s, he did the same experiment of cloning frog. Frog A, he took the egg, removed the nucleus. Frog B, he took the skin cell, now took the nucleus from it, tried to fuse these two, and that led to the development of tadpole larva. So while animal clonings were going on, there were a lot of questions were raised for the ethical issues which might be involved in doing the the animal cloning and the possibility of even doing human cloning. But there was no doubt, no question that whether the stem cells are going to be really useful for therapeutic purpose. So stem cell cloning really got more momentum. And the goal here was not to do reproduction like the human embryos, but to actually produce the stem cell for treatment and the therapeutic purpose. So just to refresh you, the stem cells, these are non-specialized cells which could actually divide and become more specialized cells if you provide the right media, right conditions, growth conditions, and they are able to replenish their own population, generate the specialized cells, which could be based on the what trigger you give, what factors you induce, it can develop into the specialized cells. So they are able to divide, capable of differentiation, and the daughter cells can actually remain in the undifferentiated state or differentiated state, depending on your trigger, your growth factors. So Dr. Shinya Yamanaka, he was the key scientist who led to the field of the stem cell cloning. And what he did, he used some transcription factors and showed that they are actually involved in the pluripotency of the cells to really changing the mature differentiated cells. And based on the various mix of the transcription factors, which was OCT3, SOX2, KLF4, MYC, he was able to demonstrate that in the mouse embryonic and stem fibroblast, one could acquire the properties which are similar to the embryonic stem cells. And these were known as induced pluripotent stem cells. So some of these iPS cells can be used for the therapeutic purpose or regenerative medicine, depending on what kind of organ requirements are there. One could try to use these specialized cells. So again, as I said, it's not a biology course, so I'm not going into too much detail about the mechanism of these, but just trying to give you the flavor that a lot of experiments, so many key findings were actually happening in the field of cloning, starting from the animal cloning and then moving on to the therapeutic cloning. So let me show you this news clip of significance of Dr. Yamanaka's work.
Next are the 2012 Nobel Prizes. The first was awarded today for groundbreaking work in reprogramming cells in the body. Ray Suarez looks at those achievements.
The Nobel Assembly at Karolinska Institute has today decided to award the Nobel Prize in Physiology or Medicine 2012 jointly to John B. Gurdon and Shinya Yamanaka.
The two scientists are from two different generations and celebrated today's announcement half a world apart. But today, they were celebrated together for their research that led to a groundbreaking understanding of how cells work. Sir John Gurdon of Cambridge University was awarded for his work in 1962. He was able to use specialized cells of frogs, like skin or intestinal cells, to generate new tadpoles and show DNA could drive the formation of all cells in the body. 40 years later, Dr. Yamanaka built on that and went further. He was able to turn mature cells back into their earliest form as primitive cells. Those cells are in many ways the equivalent of embryonic stem cells because they have the potential to develop into specialized cells for heart, liver, and other organs. Dr. Yamanaka is currently working at Kyoto University. Embryonic stem cells have had to be harvested from human embryos, a source of debate and considerable controversy.
All right, so both of these scientists, for their very original work, very basic science work, the theme of discovering that mature cells could be reprogrammed to become pluripotent. And just again, if the pluripotent term is not clear to you, this is a cell which is capable of developing into any type of a specialized cell type. So both Dr. John B. Gurdon and Dr. Shinya Yamanaka, they were awarded the Nobel Prize 2012 for this groundbreaking research, which started from 1960s to the 2008. All right, so then while this kind of research was happening, there are many scientists who were also trying to achieve more of the animal cloning success. And that's where one of the scientists, Dr. Wang Woo-suk, from the Seoul National University in South Korea. He was one of the star scientists over there who was doing many cloning experiments. And of course, in a series of papers in Nature and Science, he was showing the potential of cloning. For example, he created the genetically modified livestock of cloning. He showed a dairy cow cloning in 1999 in February. And then again, he announced cloning of a Korean cow in April of 1999. Then he showed that they could create BSE resistant cow, and then they were intending to clone Siberian tiger. So many cloning experiments were happening, and he was kind of achieving the big success in not only South Korea but in whole world of being first in showing some of these experiments. Further to that, then now they also shifted to the human embryonic stem cell research. So in addition to looking at the animal cloning, which he showed for many domestic animals, from the cows to the dogs, cats, many animals, then they also moved on to the human embryonic stem cell research. And here they were able to show that they could actually do the somatic cell nuclear transfer. And by showing this kind of a staining pattern of different markers, they were showing the characteristic cell markers for these cells, which they were able to generate by doing this experiment, which is known as somatic cell nuclear transfer. So in this case, what they did, they took the patient-specific embryonic stem cells, which were derived from the human somatic cell nuclear transfer blastocyst. And this particular study was published in Science in 2005. So again, to the groundbreaking research, which first time showed that how one could generate the immune matched human embryonic stem cells, which is actually established from the SCNT technology. And many experiments were actually done on different cell lines to establish the confidence in data. And of course, that's where the study reached to publish in journal Science. But after some time, the journal Science, reported some issues of the image problem in his paper in this particular work. And what this particular statement states that on 4 December, Wang notified the Science editors that a figure in the online material that accompanies his group's 2005 paper on the derivation of stem cells from the cloned human embryos contained duplicate image. And then from the information that we have so far, it seems that it was an honest mistake. There is no evidence that there was any scientific misconduct happened. But then some other people started finding that, they, in one paper of this group, they have reported that, one cell line was derived from 230 trials. But in second paper, they claimed that it was from the 15 trials. So a lot of discrepancy, people started, reporting in the journal. So then, after a series of these kind of reports coming forward, in December 2005, Dr. Wang Wusuk and another lead author, Dr. Gerard Scatton, they retracted this paper. They wrote to editor of the Science Journal to retract these papers. But it's not so simple that you just publish a paper somewhere and then, especially the journals like Nature and Science, and now you tell the journal that, okay, I want to retract the paper. It's not simple. So you have to now tell that why it is being retracted, what was the thing wrong, why it has happened. So now it is big concern, because especially if the groundbreaking research like this being published in Science, though then science editors are also in this scanner that how this kind of things can actually get published. So the editor themselves has to come for the press release.
The journal Science has received and reviewed the summary of the report from last night of the Seoul National University Investigating Committee that has been looking into two papers published by Dr. Wang and his colleagues in South Korea in Science in 2000 and 2004 and 2005. The findings involve serious research misconduct, finding that both papers contained fraudulent data and fraudulent conclusions. The authors of the 2005 paper had already agreed universally to a retraction. Since the 2004 paper is also implicated in these findings, we have decided to retract it editorially as well.
All right, so it became a big problem. And then the Seoul National University, there, because every university, they have the, research integrity team to look for the ethics and various kind of these scientific conduct. And all the, institutes, they have the prestige built based on these kind of, the scientific contributions. So if anything goes wrong, then the university itself starts investigation against the faculty, scientist, or student. So in this case now, Dr. Wang was under scanner that Seoul National University has constituted a committee to investigate the problems. These papers are retracted, that what all things has gone wrong, why it has gone wrong, and whether we should give a chance to this particular researcher to give justification why this thing has happened. So this was a big deal happening that time. It was in all media, whole world was kind of shocked, and whole, South Korea's, the prestige was involved because he was supposed to be a star researcher and scientist in the field. He in fact got such a big credit over there that he was given the president and prime minister, that kind of a status over there, who can travel with the anywhere in country free of thing, and all kind of amenities were given to him. So now the Seoul University is actually conducting this scientific investigation against him, but the whole country was totally with him because he had given so much proud to the country by publishing the things in big journals, and whole world was kind of astonished with South Korea's success, that public was totally involved with him very emotionally. So he has been called for investigation at 2 a.m. in the night and is still on the road. Whole country, whole of the South Korean people in Seoul, they are actually standing behind him, and they are actually waiting for him to go for the investigation. And they're very optimistic that he will come back with the clean sheet and he will be again starting his research. So this is very touching clip to show you. All right. So, reason I showed you this clip that look at the public sentiment. Even small kids, their family, they're all standing on the road in the night, waiting for Dr. Wang Wusuk to go for the investigation, and they're totally showing their faith and their confidence in him, and saying that okay, you go and you will come back. We're all with you. So whole country was kind of that this is a big conspiracy going on against him, and he should come back with the the good credit. But unfortunately, that did not happen, and this investigation revealed that both the papers had lot of scientific misconduct, and these were retracted. He was getting punishments now, and this is the Times magazine, the fallen idol from the South Korea. So now you see the kind of the punishment which he got.
Disgraced stem cell researcher Hwang Woo-seok has been sentenced to 1 year and 6 months in prison and 2 years of probation for embezzling state and private funds and buying human eggs. The Supreme Court issued the verdict Thursday, which comes 8 years after he admitted to faking his research. The ousted Seoul National University professor was indicted in 2006 on charges of fabricating the results of his human stem cell research and embezzling nearly $2 million in research funds from the government and 2 domestic companies, SK Group and Nongyap.
Well, so such a groundbreaking research, such a pride moments, the big publications, and finally he was in jail, right? Again, the whole country started showing a lot of protest, lot of support for Dr. Wang Woo-seok, and it was looking like the whole national issue that a scientist is being sentenced to jail, and that should not happen. And all of the students from the university, public, they were all kind of protesting that this kind of thing should not happen to him. Well, so I hope you got a glimpse at how much public sentiments are there for the big scientific accomplishments, and that's where the summary chart shows that how his various research work were actually published in the topmost journals, uh, and then he was sent to jail for his fabricating data, and after getting discharged from jail, again he try to make a comeback. He went to Canada to start some research over there, and he did make a small comeback when he tried to patent again for some of the cloning work, and he got a US patent from his work, which was actually quite astonishing to see his comeback.
A former leading Korean geneticist has received a U.S. patent on a controversial stem cell line that sparked a scandal nearly a decade ago. Hwang Woo-seok was earlier convicted of falsifying papers and embezzling government funds, but this latest development may open the door for his return, our Polly reports.
Disgraced cloning scientist Hwang Woo-seok is again making waves in the international scientific community with a recent U.S. patent based on his controversial stem cell research. The U.S. Patent and Trademark Office confirmed this past week that the NT1 embryonic stem cell line had been granted legal protection along with its respective laboratory process. Hwang was among the 15 inventors listed on the approved patent. The NT1 line is based on creating human embryonic stem cells by transferring the core genetic information of a normal human somatic cell into a female egg.
All right, so he really, tried to make comeback, but that did not happen. University did not give him position again. It just happened that last year I was actually in the same university, Seoul National University in South Korea, and I was invited by the president of Korean Human Proteome Organization, Professor Cho, to give a lecture over there, and he is shown in this picture on the left side. And on the right side is a building where actually Dr. Wang Woo-seok worked, and it just happened that the Department of Veterinary Science, where he used to work, is the same place here, where close to that I was there for the talk. And I must say that we did discuss about him with with the scientist over there, and what I realized that even after so many years, the kind of blot what has happened on university, and the damage which has happened on their kind of reputation, it still remains there. People are still very hurt, and it is kind of after so many years, still that particular blot remains there, and that kind of research totally got shut down in that university. So, but let's change gear. I think while this was very sad story, but something else was happening exciting in Japan, and this is a work being done by a scientist, Dr. Haruko Obokata. And given that Japanese scientists have made good credentials, especially Dr. Shinya Yamanaka, he got the Nobel Prize, and he showed so nice experiment of pluripotency. The kind of experiment Dr. Haruko Obokata was doing, people were very excited, right? And she was working at very prestigious Riken Center in Japan. So what she did, she was trying to reprogram the cell. What Dr. Yamanaka was doing, he was using some transcription factors and trying to use those to show the pluripotency. What she says that neither you need nuclear transfer nor introduction of transcription factors. You just need a simple method of stimulus-triggered acquisition of pluripotency, which is known as STAP. And for doing that, you just need to add the low pH stressors in the medium, and then that actually somehow reprograms the mammalian somatic cells and results into the generation of pluripotent cells. A simple, elegant experiment, which then she showed that how this particular stimulus triggered acquisition of pluripotency could contributing to the embryonic and the placental tissues. She did an in vivo experiment, and also then she showed that how this kind of STAP conversion could really reach out to the acquisition of not only pluripotency, but also this could reach to the ability of trophoblast differentiation. She did a lot of marker studies to show the gene patterns, and these experiments, of course, led her to publish her findings in the very, very big papers in Nature. Let's see this report of her work.
Dr. Haruko Obokata is used to working quietly in her Kobe laboratory, far away from the attention of the outside world. But today, the young researcher was suddenly plunged into the media spotlight as news of her breakthrough spread around the world. She told the collected journalists that early on, her idea was considered so bizarre, she'd nearly given up on it altogether. Nobody believed me when I told them what I'd found. So one time when I was lying awake in bed at night, I told myself that I'd scrap the experiment completely if I didn't get results the next day. Well, that next day sort of turned into 5 years. Dr. Obokata and her team have managed to regenerate stem cells from mature adult cells by immersing the tissue samples in mild acid. So far, it's only been done with mice, but if it works for human cells too, it could lead to all sorts of new treatments. In the future, this will potentially allow us to carry out organ regeneration inside the body and could help us to understand how we can control the production of stem cells to create cancer control technology. Japanese researchers have long been at the forefront of stem cell research. In 2006, another Japanese scientist was the first in the world to succeed in turning adult human cells back into stem cells. He won the Nobel Prize for his discovery. No Japanese woman has ever won a Nobel Prize so far. Rupert Wingfield-Hayes, BBC News, in Tokyo.
So of course, so Dr. Haruko Obokata's findings were quite promising, and as you just heard that Japanese scientists were actually already in the forefront of stem cell research. So her contributions were actually, people started debating that whether she can be the first women scientist from Japan who could get even a Nobel Prize. So while these things are happening, there was a lot of she was getting a big fame for her work, and her work got published in Nature, which you see this particular article of STAP, stimulus-triggered acquisition of pluripotency, and then another paper also got published, which is showing the bidirectional developmental potential of the reprogrammed cells with the acquired pluripotency. These are the papers of 2014, so it's not very old work. So this led to really, kind of big breakthroughs in the field. And Japanese media was really discussing about her, and whole world was very much proud of all her accomplishments. So let's see what Japanese media talks about her.
Meantime, some big news in biology. Japanese scientists have announced a new and faster way to make stem cells. Experts say the research could open a new era in stem cell biology. The new method involves taking cells from skin and blood and putting them in a mild acid bath until, as the researchers say, they're on the verge of death for about half an hour. Removed from the acid, the cells recover within just 3 days and come back to becoming very much like stem cells taken from human embryos. Up to now, it's taken several weeks to grow stem cells. The team is led by Dr. Haruko Obokata.
I haven't simply dreamed this up to offer a solution to some sort of single limited problem. At the front of my mind is the desire to create something that benefits all of human society, not just now, but in 10 or even 100 years' time.
Obokata has become a celebrity in Japan, but there have been complaints that the media has ignored her breakthrough to concentrate on the fact she's a woman. That coverage brought London-based writer Mayumi Tanamoto to post a blog attacking the media's handling of the discovery. We talked to her on Skype. First, we asked her to explain her criticism.
Yes, my main concern was that they put too much focus on her personality and her fashion and the way she dressed up, she decorated the lab rather than her findings. The articles which irritated me were the ones which used the buzzwords such as rikejo. Rikejo is the buzzword in.
All right, so all that, just try to show you that, she was being discussed on the media. She was actually becoming celebrity. She was given the big accolades for her work. And also there was a lot of discussion happening whether she's getting sufficient, the award for her recognition just because she is a woman working in Japan. So all this was happening, but then, the breaking news came that, okay, this particular study could be controversial. So acid bath stem cell study is under investigation, Journal Nature reported, and now the study started getting investigation. So this was kind of very shocking because this was really led to the, big credential work being done by Japanese scientists, and she was working in a very prestigious Riken center, and even her study took many years to actually materialize. But then people started, investigating her work, and in this case, some people started putting in the journal Nature that there are some issues of the figures what she is showing, and I will show you a couple of those. But let's see that now what investigation started against her.
Scientists from a leading Japanese research institute have apologized for the grave mistakes in recent papers on a new way of creating stem cells. The research was hailed as a revolutionary discovery when published in January in the British journal Nature. But mounting questions about the methodology and results forced the Riken Center to take action. NHK World's Takafumi Terui reports.
Riken scientists spent about 4 hours in front of the cameras talking about what has become an international scientific scandal. They say the findings of their investigation into the stem cell study in question were still inconclusive. They noted they've seen no evidence of deliberate data manipulation so far. But the president of Riken, who's a Nobel laureate in chemistry, acknowledged serious mistakes were made.
I would like to first and foremost express my deepest regrets that articles published in Nature by our scientists are bringing into question the credibility of the scientific community.
All right, so when they started investigating her data, there are several apparent things which came forward that, okay, this particular figure, if you look closely, it looks like a lane is pasted separately. It is not something which was originally done on a gel. Now, in this particular case, there were many times, whether these dots, red cells are real or these are kind of some artifact of staining. There things have been changed with the contrast in this case here, if you see. Now, in this particular image of her work, figure 2G and figure 1B were shown at the different panels, but this is the same image which is actually moved around to make it look like this. And then if you look at this particular GFP image of staining with microscopy and shown here is show that same thing is now increase the contrast and brightness of that. Then she tried to look at the same image whether this much part has been cropped to make it look like this. These two panels are exactly same for two different marker genes of NAT1 and FGF5. And then in this case, these were the same for different genes. And then now this particular gel was cut like this to make a panel for a different gene. And now in this case, you can see these two bands, the thick one and thin one, are actually these two only. So, like that, there are many discrepancies which were not, something one could say this is unintended mistake, just happened. It is just, manipulated data, which is somebody has tried to do all this thing to make their point. And more than that, actually, she copied a lot of text from different papers. And these are some of the articles which is in the red, shows the, how much she has already plagiarized from these articles. And it's not only one, it is from 3 different articles, right? So it just brings all the issues which we have been discussing, right? I talked to you in the last class as well about plagiarism issues, about originality, to be really, considerate, to try to be original, try to show ethics in your work. All of those issues are actually coming into the play here. And then after all this investigation, then she could not prove that it was actually done correctly. She has to come to media and apologize. All right, so you have seen that she tried to apologize. She still tried to make her case that she feels that her work, what she had done was correct. It is still she needs some more time to really repeat her experiment, replicate her experiments, and then trying to prove that these experiments have the potential. Although university gave her some chance, and they asked her to repeat her experiments, and she was actually put under CCTV surveillance for doing the experiments for 6 months' time, that whether she can reproduce her own experiments. And after given that time, she was not able to reproduce. And of course, then her papers were retracted. And again, like what you have seen for Dr. Wang Woo Suk, in this case, the staff scientist, she has become now the fall of the staff scientist in this case. After that, she opened a web page to show that she still have her hope to revive this study. And just the last line, it is my sincere wish that stem cell progress research will someday rightfully return to the forefront of scientific publication so that we all may benefit. So she was still showing a lot of hope from her research, but of course she was denied to give this kind of opportunity anymore. So I tried to convey you in terms of a biological problem of looking at the cell reprogramming. A lot of fundamental research happened in the the decades, and from 1960, Dr. John B. Gurdon for his original work on doing the frog cloning, the first time any animal cloning being done, and Dr. Shinya Yamanaka from the Japan Nobel laureate. Both of them got the the recognition for their work of doing the basic science research. Dr. Yamanaka looked at the pluripotency and the stem cell research, and both of them got Nobel Prize in 2012. Simultaneously, a lot of animal cloning work was happening in Dr. Wang Woo Suk's lab. in Seoul National University in South Korea, and Dr. Haruko Bokata's lab in Japan in Riken Center. In 2005 in Dr. Suk's case, and in 2012 to 14 in case of Dr. Haruko Bokata. And both of this work actually got so much limelight, but eventually turned out to be fake. And both of them faced a lot of punishment for the scientific misconduct which they conducted. So in summary, I just I wanted to convey you that you can always have different routes of taking your work forward, but it just takes so long time and so much patience to really be original, to really do the groundbreaking work which happened in this area of reprogramming cells, which actually received Nobel Prize. But in case of Dr. Wang Woo Suk, the success came very fast. The back-to-back different cloning experiments showed from the dogs to cows to the somatic cell nuclear transfer, variety of cloning success happened in his case, but eventually turned out to be wrong. And so as Dr. Haruko Bokata, when she claimed for the stimulus-triggered acquisition of pluripotency, or the stack phenomenon for the cell reprogramming. And again, that turned out to be a scientific misconduct case. So the biggest landmark studies have actually resulted into both the topmost prize like Nobel Prize and the topmost punishments in the areas of scientific research. So I hope you have also got a glimpse from this story that the scientific misconduct can be so detrimental, it could actually lead to severe punishments, and probably it will give you an eye-opener. While you're all starting very fresh, you're all starting very new in the new curriculum, you're trying to follow the journey of the science and research. I think it's a good idea to keep these points really in mind. To make this particular presentation for this lecture, I used several research articles where these studies were actually published, and also several video contents which I have given the keywords. If you type these in Google, you can find out these videos as well. So that's pretty much it for the presentation part. Let's now talk. Let me stop sharing my slide, and let's now talk to all of you. And I'll be happy to hear your comments about what you think, what you have learned from this particular study. Have you got any key message from this whole presentation? So I'll be happy to take Arun Prasad, your comment first. Your hand is raised there, so.
Yes, sir. So we should not manipulate any scientific data. We should present it as it is.
That's right, and it's not only just data manipulation, is actually the extreme case, but also throughout the journey, a lot of ethics and philosophy and good conduct is really required. And as we were also discussing in the last class, it just need a good practice, of developing that originality. I don't know how many of you in last 2 days actually have written 1 hour something original writing. But if you have not done that, I would like to give all of you one assignment. Just to brief a little bit, initially we thought there will be 2 lectures per week, but now it is going to be 3 lectures per week on Monday, Tuesday, and Thursday. So I will again talk to you on Monday as well. So I'd like to give you one assignment, which will be for Monday class. And that is essentially a 1-pager write-up. All of you have to write, what is your key learning from today's presentation? I'm repeating that, what have you really learned from today's presentation? I started in kind of, more scientific status of the cloning experiments, the animal cloning and the stem cell cloning, and then various ways how different type of experiments were actually even also manipulated, and what is the consequence of that. So what have you learned from this presentation in 1 page? Of course, you will not have any resource to write this particular assignment. 1 page, type that or do handwrite, and you have to email me on my email ID. So I will, I hope you all are aware of my email ID, which is Sanjeeva, SANJEEVA, @iitb.ac.in. I will put this in Moodle as well. So I'm hoping that all of you are following up the Moodle, and I'm also uploading the contents for some references, as well as the video what we talk in the class. So everything is already getting uploaded over there. Please do follow up. But I'll also write this particular assignment topic and my email ID where you have to send before the next class. So by Sunday, you have to send me this assignment. So you still get good 3 days time to work on it. All right, so let's take some more comments about, what is the key learning? What did we really, try to achieve from this particular presentation? And, what is our thought that how we should improve on this aspect? Can somebody else make comment? I think this is kind of not a very theoretical course. It will be a good idea for you to not feel afraid. And this is a good opportunity to also start communicating, right? Because as we just emphasized, you have to improve this part. You cannot be just so shy, and you have to open up. This is a good way to start discussing about your ideas. So please talk. Just give any comment or question what you have.
Sir, Karishma here.
Yes, Karishma.
So I believe some responsibility also lies with the journals. What we saw in the last few minutes is that there was some fraudulency with the images that were given in the research paper. So if the journals had investigated it properly before publishing, the whole thing had not happened at the first place. They could have checked the gel images and they could have easily identified that something had been cut and pasted there. They could have easily identified the other images and see that it has been some way rotated and it's the same image. So that could have been investigated at the initial stage before they have given the acceptance for publishing.
I totally agree with you, Karishma. So this is actually a very good point, and I'm just trying to again go back to that paper and open this particular. Let me share my screen again for a minute for this discussion. So I hope you can see my PPT again. So if you look at this particular paper, right, the authors and investigators who are involved here. So people from the Riken Center, then another institutes of other departments of Riken. Then in Kansas, US, some scientists are there, and several other medical university of Tokyo, Yamanashi University here, and then in another paper, Riken, Genome Reprogramming Center again, Tissue Engineering Center of Harvard Medical School, Boston, USA, various centers of Japan, Kobe and Yamanashi, etc. So what is the problem here is many of these scientists are actually who are part of this particular work, they're very accomplished in their field. They have been already publishing. So even the scientist from Harvard, scientist from Japan, she is the quite young investigator in that way. And she was working along with a team of the big scientists. Now, the problem here is not everybody was fraud and not everybody wanted to publish this particular thing fraudulent way. It just happened that one of the scientists like Harusho Obokata, she became greedy and she started manipulating data. She started presenting to all of these people, it's actually happening very reproducibly. And that's where there is a lot of trust involved in science and research, right? And when a paper goes to journal Nature with all these kind of big names of the people involved who have already been publishing in that field quite well, then they don't, think about this level of low level of scientific misconduct. That was the problem in this case. But journals have really learned a lot from this study and these problems of scientific misconduct. And now all the journals have the first thing is to check for the plagiarism. As I told last time also, sometimes your paper will go to a journal and it will come back in 2 days time. You have no idea why it came back so fast. And the main reason first could be that your text itself was totally copied. So the editorial team itself even did not send to editor for even looking at that. Paper right away got sent back. Then after that, then the editors who are actually involved, now they have also started keeping the people of the image specialization, the people who can actually look at images well and use software to ensure that images are not manipulated. And there is a very fine, delicate sometime in presenting the scientific data that to all the big journals you see the data looks so fancy, so nice, very well, beautifully presented. So you all want to also present your work so elegantly, but while doing that, it should not be changed. I think that's the important thing. So if at all for representation purpose you are cropping something, you are putting a small panel from here and there and you are showing all that together, you can still put all the actual original images in the supplementary data. And then you can refer that these are the for representation taken from different panels, but the original images of this data is actually put in the supplementary data set of the paper. So now when anybody want to look at the original images without any change of the contrast and image and brightness, that's all available. That's not a manipulation then. You are only representing something to make a kind of decent case. But you are providing the original images. That's really important. Otherwise, now the journals have learned again this kind of mistake. What has happened? Now they also have the people in their editorial team who are looking at the scientific images. So the writing part is of course the standard thing. The scientific plagiarism check happens first thing. But after that, also the many journals which deals with a lot of images, cell-based journals, they look at the images very carefully before even they again do peer review process. And a lot of papers have actually been retracted in the overall field based on image manipulation. That is actually one of the very gray area for things to actually happen. In this case, of course, there were many misconduct, many issues happened. But this is actually a good point to bring in that journals has to also become more careful. And that's where the journals like Nature and Science and Cell and all the big journals who publish very highly, with the high impact factor, with the very valuable findings, they also see large number of retractions happening. And that is just that, because when somebody publishes a work in Science or Nature, there will be, 1,000 people who are following that work. Many people would like to follow their work in their own lab. And they would like to reproduce that work and see, okay, can I also get the same data? What is step is so simple. You take a small drop of acid and put in the stem cell, and now it is, acquiring the pluripotency. How fancy it is, right? Why don't I try the experiment in my lab today? But that did not work out. Then, okay, let me try it again and, follow the protocol more rigorously, how the authors have described. And then I did the same thing. I'm not finding it working well. Then I will go back to the paper and say, okay, how they have presented their data. And then I observed, okay, now this thing looks like something wrong. This image does not look right. Then the text of that, okay, how they found this methodology, okay, I checked that method. And now you go back and search in Google or other kind of, the plagiarism software, and you find this particular methodology was reported exactly same in 2 other papers. So this is how the things start coming back. When you publish highly in the top journals, it also, opens up the, areas for a lot of discussions when people want to reproduce that work of the high significance. Having said that, I hope all of you learned that, the big scientific discovery which is happening, takes a lot of time, a lot of patience, and it cannot be just, expedited in this manner. And many times it actually has led to these kind of scientific misconduct. I just showed you one story in kind of, which because I was following it, it was of our field. And it just happened that cloning, when I was growing up in this field of science as undergrad and, wanted to do research as my career, the cloning was happening a big thing at that time of, the sheep being cloned and human cloning. And then I was following very closely the work of Dr. Huang Woo-Suk for different animal clones. And then the stem cell cloning work and the Nobel Prizes were given. So this entire thing, as it like happening in front of you. So then you start seeing that what happened to these research and what happened to these researchers. But this is not the only case study which is actually happening. There are many studies like that. There are many problems that happen, not only in biology, but also in physics, also in the computer science. Many fields, many type of scientific misconduct has happened. So I think it's really important that, while all of you are really gearing up for the research of the career, it's actually important to learn the best practices from the beginning. And I think the communication skill course do provide you a very good opportunity to open your eyes, to think about how best you can start improving yourself. Not that you would like to do any of this thing, but it requires just so much practice of improving your overall quality, whether it's writing, your presentations, your original thinking, your way of learning about ethics and philosophy of doing research. All of that requires just tremendous amount of effort, right? It can't be just achieved in a single day. And what was also, kind of very alarming to see, which is the reality that the prestigious institute, like, the Seoul National University in the first case and Riken Center in the second case, when they see any kind of misconduct being happening, then they conducted, the full investigations. And because their own university's prestige is also involved, So as soon as you become the, you get enrolled in the institution, all of you, many of you would have just joined IT Bombay now and not even come to the campus. But now you have a brand of IT Bombay, right? So anything which you do actually belongs to IT Bombay. Your good and bad both belongs to IT Bombay. So I think that's where now all of a sudden you just came from a college, you were studying somewhere, and now you got admission to IT Bombay. Now you became a student, a property of IT Bombay, and your every conduct, every particular property, your success, everything, whatever you do, actually becomes part of the university's reputation. So of course it doesn't, did not take you very long time to finish your graduation and coming to the IT Bombay, and that duration is very short. It cannot change your entire learning right away. But I think it's a good idea to immediately start practicing. And it's not only IT Bombay, every university wants to maintain their prestige. Every university wants to maintain their level of credit, their credentials in the field. So it's a good idea that when you are representing now any institute, your success is also going to determine the institution's success. This is where I think it's really important to take these kind of courses and learn the best practices from the variety of faculty. And this is why this course is actually going to be the blend from different faculty members who are going to talk to you about different skills required for scientific communication as well as doing good research work. I hope today's story will remain with you for a long time. Whenever you are deviating from the path, whenever you are want to take any shortcut, you will remember the story, and that will prevent you from going anywhere. And I'm hoping also that you would take at least some tips which we are sharing with you with over the experience of a struggle, over the experience of work, that do make the effort to improve these skills. It does take time, but with efforts, these can be acquired easily. Everybody cannot be gifted writer, everybody cannot be gifted presenter, but all of you can acquire very, very professional skills of doing these activities. And that's where I think you have to make continuous effort. Every presentation needs some practice, lot of research work, lot of groundwork of making that presentation. And every writer requires a lot of thought process about writing originally. So please do start making effort in both of these directions. I think this will really give you a lot of lead for the long term. Anybody want to add any comment? Please do that. Otherwise, I'll stop this call shortly. Any comments? Anyone has any questions or comments? All right. If not, I know that you may have another class. It's already 10:32. So I'll stop this call. I will put this recording also in the Moodle along with the assignment topic. Please do write originally. Do try to show your best. When I read 50, 60 write-up, I should not feel it's a waste of my time. I should feel that, okay, students have really made effort and they have tried to at least learn some tips what I have given them to try to be original and write as coherently as possible. So please do try to make a good effort of improving. This could be the first step of you to improve yourself in this scientific journey. Thank you very much.